All right, let's jump right in. Today, we're talking about one of the most incredible achievements in all of modern medicine, organ transplantation. It's this amazing story of generosity, cutting-edge science, and a really delicate dance with our own biology. But for every miracle, there's always a challenge. And for transplantation, that challenge really starts with a single, pretty stark number.
So what's that number? Well, it's 4,000. That's roughly how many patients in the UK are waiting for a kidney transplant at any given time. And remember, that's just one organ in one country. The shortage is a global problem for hearts, lungs, livers, you name it. It creates these waiting times that can just stretch on for years. And this huge need is really the engine driving all the science we're about to get into.
So this is the million-dollar question, isn't it? How do you take a living, complex organ from one person and convince another person's body not just to accept it, but to actually make it its own? To figure that out, we're going to follow the organ on its incredible journey. First up, where do these life-saving gifts even come from? Then, how in the world do we keep them alive in a crazy race against time? And finally, what happens when we introduce this new organ to its new home? A body that has a powerful, built-in security system ready to attack.
Okay, let's start right at the very beginning of this journey. The gift of an organ. It mostly comes from two incredibly generous sources, deceased donors and, believe it or not, living donors. And each of these paths has its own unique set of scientific challenges and, of course, advantages.
Now, when we talk about deceased donation, it's not just one thing. There's a really crucial difference. On one hand, you've got donation after brainstem death, or DBD. In this situation, even after death is declared, the donor is kept on a ventilator. This keeps blood circulating, feeding the organs with oxygen right up until they're recovered. But on the other hand, you have donation after circulatory death, or DCD. Here, circulation stops before the recovery begins. This means the organs go through a period without any blood flow, a really critical state called warm ischemia, where the tissue is literally starving for oxygen at body temperature.
And that brings us to living donation, which is a whole different ballgame with some massive advantages for the recipient. First off, the organ is coming from a healthy person who's been checked out from top to bottom. The surgery itself is planned. It's an elective procedure, not some emergency scramble. What this means is that the time the organ spends outside a body, in cold storage, what we call the cold ischemia time, is super short. And the result of all that? Just way better long-term outcomes for the transplant. But, and this is a big but, with all those advantages comes this really profound ethical complexity. This quote right here really cuts to the heart of it. A perfectly healthy person is going through a major, life-altering surgery with all its own risks, not for their own health, but for someone else's. It's just an incredible act of altruism, and it really highlights the immense responsibility and care that has to surround this entire process.
So, once an organ is generously donated, the clock starts ticking. Immediately. The very next part of its journey is this frantic race against time, where science has to step in and basically create an artificial bubble to keep that organ viable until it can get to its new home.
It's a fascinating question, right? When you think about it, without oxygen, our cells just can't do their normal energy-producing thing. They have to switch to a way less efficient backup process. And when that happens, energy stores just plummet, nasty waste products like lactate build up, and the cells themselves start to swell and just break down. So the mission is crystal clear. We have to stop this whole process of decay, dead in its tracks.
For decades, the gold standard for this has been something called static cold storage. And it really boils down to three key ideas. First, you flush all the blood out of the organ's tiny little blood vessels so it doesn't clot up. Second, you get it cold and fast, down to below 5 degrees Celsius. This deep chill dramatically slows down its metabolism, so it barely needs any oxygen. And third, you replace all the normal body fluids with a special ice-cold preservation solution that's designed to protect the cells.
But of course, science never just stands still. The old organ on ice method is evolving into something way more dynamic. These incredible machines that can not only preserve organs, but actually restore them. Things like hypothermic machine perfusion are constantly pumping cold fluid through the organ. And even cooler is normothermic machine perfusion, which basically puts the organ on a mini heart-lung bypass machine, circulating warm, oxygenated, blood-based fluid. This is a total game changer. It's not just about hitting pause on decay, it's about actively reconditioning the organ. We can now take organs that we might have considered too marginal before and make them viable. It's expanding the entire donor pool.
Okay, the organ has been donated, it's been preserved, and it's been successfully implanted. The surgery is over. But honestly, The biggest challenge is just getting started. And it's a battle that happens on a microscopic scale. The recipient's body has this incredibly sophisticated security system, the immune system, and its number one job is to find and destroy anything it doesn't recognize as self. So how does this security system actually spot an intruder? Well, it all comes down to these molecules right here, human leukocyte antigens, or HLAs. The best way to think of them is like a unique molecular fingerprint on the surface of all of your cells. And because our genes for these are so variable, a donor's HLA fingerprint will almost always look different from the recipient's, and it's that mismatch that sounds the alarm.
The immune response that leads to organ rejection is this super coordinated multi-step process, and it all kicks off with what we call signal 1. This is that first moment of recognition. A recipient's T cell, which is like a key soldier of the immune system, literally bumps into the donor's foreign HLA fingerprint. That's it. That's the initial intruder alert. The moment the security system realizes, hey, something here is not right.
But just one alert isn't enough to launch a full-scale attack. The system needs confirmation, and that's signal 2. This is what's called a co-stimulatory signal, a second, confirming handshake between the T cell and the cell showing that foreign fingerprint. You can honestly think of it like two-factor authentication for the immune system. Without this confirmation step, the attack just doesn't go forward.
So once signal 1 and signal 2 have both been received, that T cell gets its final orders. This is signal 3, and it's driven by a growth factor called interleukin-2, or IL-2. This signal is the call to arms. It tells the now fully activated T cells to start multiplying like crazy, creating a massive army of killer cells, all of them specifically programmed to hunt down and destroy that new organ.
Okay, so if the body is literally hardwired to attack the very thing that's supposed to save it, how can transplantation possibly work? Well, the answer lies in pharmacology. We have to intentionally, and very carefully, weaken the body's security system. This is the art of immunosuppression, basically declaring a truce in that battle within.
This right here illustrates the incredible balancing act that's at the very heart of immunosuppression. The goal is not to completely obliterate the immune system. I mean, if you did that, the body would be left totally defenseless against a common cold, infections, or even cancer. The real challenge is to dial it down just enough to stop the organ rejection while still leaving the patient with enough of a defense to survive in the world.
So to pull off this balance, doctors have a whole toolkit of powerful drugs that get in the way of that three-signal process we just talked about. Calcineurin inhibitors are a real workhorse. They block the production of that call-to-arms molecule, IL-2. Then you've got antiproliferatives, which stop the immune cells from copying their DNA so that army can't grow. And corticosteroids are just potent, broad-spectrum anti-inflammatory agents.
And one of the really key innovations in this field is a class of drugs called mTOR inhibitors. They work in a really clever way. Instead of just blocking a signal, they mess with the cell's internal machinery directly, basically arresting the T cell's division cycle. And a huge advantage is that, unlike some of the other frontline drugs, they aren't toxic to the kidneys, which, as you can imagine, is a massive, massive benefit for patients.
So, as we've seen, the true miracle of transplantation goes way, way beyond the incredible skill of a surgeon in an operating room. The real, ongoing challenge is a lifelong negotiation. It's all about managing this delicate, chemically induced truce between the life-saving gift of an organ and the body's own powerful guardian, the immune system, which, at the end of the day, is only trying to do the job it was designed for. It's a battle and a partnership that has to continue for the rest of a patient's life.